Welcome. Today, we're diving into container closure integrity testing, or CCIT, for sterile products. I'm Dr. S.K. Singh, and together we'll explore why ensuring the seal on sterile containers is critical, not just for safety, but for product quality and patient trust. Let's get started. Let's start by exploring what container closure integrity testing, or CCIT, really means. It's a way to check that a sterile product's container and its closure keep out microbes, moisture, and oxygen throughout the product's entire life. The goal is clear: maintain sterility, protect product stability, and meet strict regulatory standards. This testing is especially important for sensitive products like injectables, vaccines, biologics, and prefilled syringes, where any breach could compromise safety or effectiveness. When we talk about global regulatory requirements for sterile product packaging, several key standards come into play. USB chapters, like 1207 and its subsections, guide us on evaluating package integrity and selecting the right test methods. The EU's GMP Annex 1, updated in 2022, pushes for deterministic container closure integrity testing whenever possible. Meanwhile, the FDA offers guidance specifically for container closure systems in drugs and biologics. And on top of that, international bodies like ICH and WHO provide additional frameworks to ensure packaging keeps sterile products safe and effective worldwide. When integrity fails, the risks can quickly multiply. Microbial contamination becomes a real threat, wiping out any confidence in sterility. Moisture getting in can ruin lyophilized products, while oxygen ingress might oxidize sensitive active ingredients. Product leakage isn't just a waste, it risks dose accuracy and patient safety. And on top of all that, regulatory bodies take notice, leading to recalls, warning letters, and a flood of market complaints. These risks show why maintaining integrity is absolutely critical. Let's take a closer look at the different types of container closure systems we work with. Cartridges, for example, use a rubber stopper and a crimp seal. Each system faces integrity challenges, things like seal defects, cracks, pinholes, or incomplete crimping. Glass vials combine a rubber stopper with an aluminum seal, while ampules rely on a glass fusion seal. Prefilled syringes include a plunger and needle shield. Blow-fill seal containers stand out with their integrated closure system, designed to maintain sterility throughout. Let's talk about deterministic methods for container closure integrity testing. These include vacuum decay, pressure decay, helium leak detection, high-voltage leak detection, and laser-based headspace analysis. These methods are preferred by regulators because they provide objective, reliable results. On the other hand, traditional microbial ingress and dye ingress tests are more subjective, take longer, and often have lower sensitivity. Plus, dye ingress tests can be destructive. Understanding these differences helps us choose the right approach for ensuring product safety. Let's talk about vacuum decay technology, a method that tests package integrity by applying a vacuum around a sealed container. If there's a leak, gas moves, causing a pressure change we can detect. It's non-destructive, so the product stays intact. It's also quantitative, highly sensitive, and can be automated for efficiency. This technology typically detects leaks as small as half a micron up to 10 microns. You'll find it used in vials, syringes, and blow-fill seal products. Let's talk about high-voltage leak detection, or HVLD. This method works by applying an electrical voltage to liquid-filled, non-conductive containers. When current passes through the liquid, any defect changes how electricity flows. The instrument picks up on that change, signaling a leak. HVLD is especially good for injectable solutions, biologics, and ophthalmic products. It's fast, non-destructive, and can inspect every single unit, making it a powerful tool for quality assurance. Let's talk about helium leak detection, a crucial process in many industries. Helium is an ideal tracer gas because it's inert, non-toxic, and has tiny molecules that slip through even the smallest leaks. By introducing helium into a system and using sensitive detectors, we can pinpoint leaks that would otherwise go unnoticed. This method helps ensure safety, maintain efficiency, and prevent costly failures down the line. It's a precise way to keep things sealed tight. Here, we see the result of our test. The pressure difference measured was 5 millibars, well below the 10 millibar threshold. This tells us something important. The container closure integrity was maintained throughout the process. In other words, the seal held strong. This slide also points to a calculation example using vacuum decay, showing how we arrive at these numbers and confirm the container's reliability. It's a clear, quantifiable way to ensure quality. When we talk about method validation, there are key parameters we need to focus on. Sensitivity and specificity tell us how well the method detects what it's supposed to, without false alarms. Accuracy and precision show us how close and consistent the results are. Repeatability and reproducibility ensure the method works reliably over time and across different conditions. We also consider the detection limit, the smallest defect we can confidently identify. To test all this, we use positive controls, artificial defects made with laser-drilled holes, capillary tubes, or microdefects, typically sized at 1, 5, or 10 microns. Let's talk about how we monitor container closure integrity, or CCIT, throughout a product's life cycle. We start with the initial release, making sure everything's sealed tight. Then, during the stability program, we check at multiple points, 0, 3, 6, 9, 12, and even 24 months. We also test after transportation, simulating shipping conditions, and after any packaging changes, like modifying the closure. For example, a lyophilized vaccine stored at 25 degrees Celsius and 60% humidity has its CCIT tracked continuously throughout its shelf life. When regulators come knocking, they focus on several key areas. They want to see thorough CCIT validation reports, solid defect challenge studies, clear evidence that methods are suitable, reliable stability data, and consistent trending reports. Common pitfalls: relying only on die ingress tests, skipping proper validation, missing defect challenge studies, and neglecting life cycle monitoring. Annex 1 raises the bar. It expects a strong scientific justification for the chosen CCIT methodology. This slide highlights where we need to be sharp and prepared. To wrap up, we've drawn on guidance from key organizations like the United States Pharmacopoeia, the FDA, and the European Medicines Agency. At the heart of today's topic is CCIT, a vital tool for ensuring sterility in pharmaceutical products. We've seen how modern deterministic methods, such as vacuum decay and helium leak detection, offer greater sensitivity than traditional approaches. Validation is essential to prove these methods catch critical defects reliably. And finally, integrating CCIT throughout a product's life cycle helps reduce contamination risks, regulatory issues, and costly recalls. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you.